Bismillahirrahmanirrahim. I welcome you in Pakistan International School Thai virtual learning system. Dear students, Assalamu alaikum wa rahmatullahi wa barakatuhu. I hope you all are enjoying your good health at your home. Dear students, this is biology class for second year students, as you know, and in this lecture, we are studying chapter number 23, and that is the continuation of the previous topic that was gene expression. In gene expression, we have talked about the process transcription, and in today we will talk about very important mm, like the next topic of that is genetic code as well as translation. So let's get started. As you know that gene expression, we have talked about gene expression, that the DNA, the genes present in the DNA, they transfer their messages in the form of protein, which are required for the body metabolic activities. And then that it represents in the form of a phenotype, any type of a phenotype. So DNA transcribed into RNA, messenger RNA, and other RNAs, and by the help of ribosomes, they make the proteins in their required form. So in the previous topic, previous lecture, we have talked about the journey of DNA up to messenger RNA. So now we are gonna talk about, this is, sorry, messenger RNA. Now we are gonna talk about the next step of that translation. But before going to that topic, we will talk about very important, which is related to the translation and protein synthesis, that is genetic code. Yes, students, genetic code, the term was given by George Gamow, and he postulated that three-letter code must be employed to encode the 20 standard amino acids used by living cells to build proteins. So as we know that we are having four letters A, G, T, and C, it means four base pairs for nucleotides which are found in the DNA. So they are organized in the form of codons, like in the form of proper sequences. So the collection of codons is called actually the genetic code. And almost 20 amino acids are there, and there should be for 20 amino acids 20 codons. So because three if like three base pairs form a codon, so there must be 20 codons for making 20 amino acids. So each codon could have three nucleotides to impart specificity of each amino acids. So if one nucleotide is there, before the scientists were just searching about that, if one nucleotide is there, so how many combination can we make? Only four combinations, because there are just four base pairs over there. So two nucleotides, so we can have the 16 combinations. But if we are having three nucleotides, so we can have 64 combinations of, and that 64 means more than 20 amino acids. So different amino acids can be formed from there. So if we see genetic code in detail, so we can see this is the first base codon, second base codon, and third base codon. So we multiply all of them together, and then we will find all these type of genetic code. These are the named is called like named of amino acids that these are all amino acids which are like define these type of codes. How these codes are formed we'll talk about in a few seconds. So there are only 20 amino acids that exist, and actually 64 possible transfer RNAs molecules are there. So 64 possible transfer RNA molecules are there. So every transfer RNA carries one codon, one anticodon against the codon which is present on messenger RNA. So if there are four, so 4 and 4, they are multiplied and it will be 64 possible combinations. How they are, we'll just talk about this. So for example, if there is GCU, so that is codon for glycine amino acid. This is the first one, this is the second one, and this is the third one. So here we can see G and C and U. So we can have this type of codon, and this can be the glycine. So these on messenger RNA, there are the triplets. Triplets are called codon. The triplets, a sequence of these nucleotides, nitrogenous bases. We can say the GCU, ACU, AAG, CUG. These are cytosine, uracil, guanine, and adenine, because the uracil is replacing thymine in messenger RNA, thymine in the DNA. So here you can understand properly that is DNA codon and that is RNA codon. That is also called cryptogram. Cryptogram. So these are all TCAG, TCAG, and TCAG. Like it is defined, this cryptogram is defined like that, that all 64 possible combinations can be formed. So here, if we multiply this T with this T and with this T, so we can get T, T, T. This T with this T and with this C. So that means TTC, this T with this T and this A, so that will be TTA, and this T with this G, so that will be TTG. So TTT and TTC, that is for phenylene, and that is TTA, TTG, that is for leucine. So there can be like different amino acids. So like more than one codon can be the required codon for the amino acid. So like these are codons, they can also form this type of amino acid. So more than one type of codons can like make amino acids, a single type of amino acids. So these are the DNA. So here the thymine is replaced with U. So you can see here, this is GCTT. So here is UU. So UUU, it is phenylene and leucine, like this methionine, valine, alanine, threonine, so serine. These are all like amino acids. Amino acids by which are formed by different combinations of genetic codes. So here you can see some changes, like these two and this one, they are called stop codon. So there are 3 stop codons in a messenger RNA. How it will work, we will just talk about in a few seconds. So the triplet sequence of messenger RNA that specifies certain amino acids, that is called genetic code, and almost 64 different type of combinations of bases can be formed. So 61 of them are they code for 20 amino acids, but last three codons like UAG, UGA, and UAA, which I have just shown you before, they do not code for any type of amino acid. So they are the termination codons, which we'll talk about in like the translation in the last step of the termination. So they also degenerate. It means more than one triplet codon can specify same amino acid, as we have seen in this diagram, like more than one type of like triplet codons can code for the same amino acid. Genetic code can also be like we can say that is the sequence of base pairs, and the sequence of codons in the messenger RNA defines the primary structure of final protein. What type of codons are sequenced in messenger RNA, then it will determine what type of uh, that is amino acids will be attached in the polypeptide chain. So three nucleotides in messenger RNA that will form a codon. So here you can see that is, for example, messenger RNA, because there is U instead of T. So that is the indication that this is messenger RNA, as well as it is single-stranded. Now the double-stranded DNA is double-stranded. So AUG, that is codon 1, like this one, these three. ACG, this is another codon. GAG, that is another codon. And like this, they form different type of codons, different combinations of codons. So codons are of different types, like there are some sense codons and signal codons. So signal codons are, we can say they are just indication of initiation or starting of the process of that is translation, as well as stop codons or termination codons are there. If we talk about sense codons, so in sense codons, they are the codons that code for amino acids, like they form the protein, they are called sense codons. And if they are not coding for the proteins, so it for amino acids, so it means they are non-sense codons, or they are called signal codons. And these codons are like signals for during protein synthesis. They have to start or they have to stop over there. So they can be AUG, UAA, AUG, and UGA. So there are two type of signaling codons, like termination and initiation codon. So termination codons are UAA, UAG, and UGA, and they are also called nonsense codons. We have talked about before that one, and that referred to as amber, ochre, and opal codons. They are, that's very interesting names for those codons. And if we talk about initiation codon, so AUG is initiation codon, and it codes for the first amino acid, that is methionine. And that methionine in eukaryotes and formyl methionine in prokaryotes. Against these codons, there is anticodons, and that is present on transfer RNA, which when they form the complementary pairing with the codon, so then there will be attachment of amino acid. So for example, this is a transfer RNA, so it is having three um, sequence of base pairs, like that can be A, G, or G. So that is anticodon against U, because complementary of U is A, and here is C, and here is C. So that AGG and UCC, like that is codon which is present on messenger RNA, and that AGG that is present on transfer RNA. So codons are present on transfer RNA against the codons which are present on messenger RNA. So the third position of this one, that is vowel position, it means it can be changeable, like against the last codon, the last base pair which is present in the messenger RNA. So there are some characteristics of genetic code, like there is universality, like genetic codes are universal. They are present commonly in most of the places, like AUG is the codon for methionine in mitochondria, but same codon AUG codes for isoleucine in cytoplasm. So with some exceptions, genetic codes are universal. And also we can take an example of that codes for arginine in bacteria, in human, in all other organisms where genetic code has been studied. So AGA is specified for, that is arginine. That is for arginine for every living organism. So next we will talk about non-ambiguous, like genetic code is non-ambiguous. Thus one code cannot specify more than one amino acid. Sometimes it happens that every codon is having its different amino acid. So they are also non-overlapping, like one base cannot participate in the formation of more than one codon, and that is called non-overlapping. For example, if there is the statement like sequence A, U, G, A, G, C, G, C, A. So how will we read it? We will read it AUG, AGC, and GCA. We will not read it AUG, then the other one UGA, and the other one GA. This is a wrong condition to read these um, codes, because that is not possible. This is not possible. So we have to read just three sequences in one code, then other three in the next code. So they are non-overlapping. It means this sequence is separate for itself. Then it is a continuous translation, transcribed and translated continuously from a fixed starting point, and like stop at the fixed point, like their genetic codes for initiation and for termination that is defined. Other is that there is no punctuation between these codes, like AUG, there is no punctuation. We just write AUG for that. Genetic codes has also polarity, like the code can be a definite direction for reading a message. For example, we are reading UUG, that stands for leucine. So if we just go this way, GUU, so that stands for viline. So it means they are different. So they are also having polarity, one side or the other side. Degeneracy of genetic code is also very important characteristic of that amino acids can be coded for more than one codon. For example, we can say UUU and UUC, that it codes for phenylalanine, and these four and these two, that's for leucine, and these are like more than one codon can be coded for one amino acid. Let's talk about very important step of gene expression, and the last one also, that is translation. Like the message of messenger RNA now is going to translate it in the form of protein. So the transcription process done in nucleus, we have talked about that messenger RNA goes from post transcriptional modifications, and after that it will move towards the cytoplasm of where ribosomes and transfer RNAs all that are waiting for that to just prepare protein, and so that is done in cytoplasm. So translation is actually the process by which ribosomes read the genetic message in messenger RNA and produce a protein product according to the message's instructions. So here we can see these are the transfer RNAs. They are having amino acids over it. This is the largest subunit of ribosome. This is the smaller subunit of ribosomes, and messenger RNA is attached to this smaller unit of ribosomes. Here are some sites of ribosomes, A, P, and E side. So we'll talk about in a few seconds. And then on the P side, these protein chain is going to be elongated and prepared over there. So for translation, there are some things needed for that, like ribosomes, RNAs, amino acids, initiation, termination, elongation factors, like these are enzymes and proteins, which are required for that, some energy sources. And very important, there is the process before initiation, that is activation of amino acid. Activation of amino acids before starting the initiation of that translation process, amino acids have to be activated because amino acids are present in the cytoplasm all the time, so they are not in active state. So for to make them activate, they have to be modified. So what happened, each transfer RNA binds with a specific amino acid. So this is a transfer RNA, this one. It has to be bound with specific amino acids before it has to pass on through some changes. Amino acid is catalyzed by the specific transfer RNA activating enzyme that is called amino acyl transfer RNA synthase. That is the enzyme which helps to perform and this function. So amino acid is going to attach at the 3 and 3' end of transfer RNA, and there is a complex formed with amino acyl transfer RNA complex um, with the help of ATP. Energy is required for that phosphate. 2 phosphate groups are detached from that, and in the next AMP is attached with that, so that AMP is required to attach to give the bond to the specific transfer RNA. And after that, that amino acid is attached with this transfer RNA, and AMP is released from that. That is called amino acyl transfer RNA complex, which is the charged and activated now. The first step of that that initiation complex formation, so that involves the first joining of messenger RNA with the smaller ribosomal subunit. So we know that in eukaryotes, that is 80 Selberg, like the larger one is 60 and the smaller one is 40. So it will make 80 S of a ribosome, and in prokaryotes that is 70. So in eukaryotes, what will happen, this messenger RNA will bind with smaller subunit of that ribosome, and also the initiator methionine transfer RNA. So this initiator transfer RNA, which was modified, which we have seen in the previous slide, that bind with methionine. Methionine is the very first amino acid, or we can say the starting. It is having the starting codon, and it is having, like, methionine is having this starting codon here, or anticodon, and for that there is a methionine amino acid. So this is the starting for every type of protein polypeptide chains. So initiation factor is needed for there, that is like additional proteins, or that is the enzyme that is called initiation factor. This binding is controlled by an enzyme called initiation factor, and the same time a 5 prime end of messenger RNA also binds to the smaller subunit of a ribosome. This one will bind first with the smaller ribosome, that 5 prime end. Then initiation factor transfer RNA will also be joined here, where at which site, that is the P site. And this is, like, we can see AUG is the starting codon and complementary codon on UAC, in which methionine is attached. So that methionine is called N-formyl methionine. Then there is needed an energy compound, that is GTP, that is like changed into GDP, and that phosphate group is released, and what we can say, like, energy is utilized to bind larger subunit. Also in this ribosome, the larger subunit, there are 3 sites: A, P site, this is P site, and E site. What are these sites? So here in the diagram, you can see the formation of initiation complex. Look, this is for prokaryotic cell, in which 50 and 57 bar, this is the larger subunit is there, but in eukaryote that is 67 bar. So this is the smaller one. In that first codon AUG, so this transfer RNA is attached, then ribosome is attached, so the information of initiation complex is completed here. So ribosomes are having 3 sites, A, P, and E. That P sites is actually called as amino acid entry, or that is called amino acyl site, that is also called binding site, in which amino acids going to be bind with one another and form the polypeptide chain. That is that is A site, that is called amino acyl site, in which we can say the transfer RNA comes and bind over there, and it move towards the next site, that is called P site, that is peptidyl transfer RNA. Here the transfer RNA leaves its amino acid, and peptide bond is formed, that is called binding site also. And E, then our transfer RNA moves towards exit site, and from here that that transfer RNA exit from this side, exit site. So here now we'll see polypeptide elongation, so that having 3 steps: codon recognition, and that is also like happened when whichever codon or messenger RNA is exposed to A site. This is the A site. It's a anticodon bearing amino acyl transfer RNA complex binds it with the help of enzyme that is called elongation factor. Here elongation factor or enzyme is like utilized for binding this transfer RNA here, and as well as hydrolysis of GTP occurs and energy is utilized here. Then in the next step, this codon which was present here, it will move toward the next side. You can see peptide bond formation. Now transfer RNA molecule and larger subunit catalyzes the formation of peptide bond here. Here the peptide bonds are going to be formed. Now you can see in the next step, peptide bond is formed like this chain is moved towards this side. Like here, all the bonds, all like polypeptide chain will make the bond with this A side. Now, and the next step, translocation, like this. Transfer RNA will shift towards exit side. From here, it will move out. And then because this transfer RNA, uh, ribosomes will move slightly towards this side on transfer RNA from 5 to 3 prime end. So here you can see elongation, polypeptide elongation of that. So this will move towards, like here, another transfer RNA is going to be attached. Here the peptide bonds are going to be formed. Then this will shift its peptide bonds here, and then it will move outwards towards, here is E side. Actually, it will move from here towards E side, and then they will move out of this. Out of this ribosomes. So you can see a messenger RNA is not moving. Yes, this is moving over there with the codon. There is anticodon formation, attachment over there, and look here once again. Here the peptide bond is formed with the help of enzyme. That is enzyme is peptidyl transferase, because this peptides will transfer here with arginine, valine, and methionine, and then it will shift towards the next site and it will move out. So this is how the polypeptide elongation occurs. So after these steps, when they are repeated again and again, there is the stop codon occurs and termination will be happen. Termination will not be happen without codons. The codons on messenger RNA UAA, UAG, and UGA, it will come when it will come on the A side. So there is no transfer RNA which is having anticodon against them, because they don't, as we have seen before, they don't form anticodon, but they are stopping codon. So when they come here, so there is no transfer RNA for that, but there is a release factor. That is a protein which comes here and binds with, you can see here, binds with messenger RNA codon, and from here the last transfer RNA is released, and from here the polypeptide chain gets free from here, and this is the required protein. And after that, releasing the larger subunits, transfer RNA killing peptide chain is also released. So all the parts of this translation process will be disarranged and dispersed in the cytoplasm. So here you can see the termination process. Like from P, it will move towards this one. Then a UGA, this is we can say the stop codon. And here another factor will come, that is called termination factor, also a release factor. So this will move up, and from here, now we can see now this one. This release factor will attach here, and from here the polypeptide chain will move on, will be separated. All the parts of that complex will be disassembled. Just see once again. Here UGA, that is the stopping codon, release factor just join here. From here polypeptide chain will removed, and all the assembled part will be removed of this effector. So, dear students, I hope you understood it. That was very interesting. With that, we have completed gene expression. So, we have found now our required protein, which is in the linear structure. But before it has to be functional for any type of metabolic activity, it has to be go through conformational changes. Like there can be secondary, tertiary, or quaternary structural changes can be happen in the proteins. So then it will be activated, and we are actually dependent on different types of proteins, like in the form of hormones, antibodies, antigens, and enzymes, and all the body functions are held with the help of proteins. So I hope you found it very interesting, as well as you got lots of information. Wish you all the best. Inshallah, we will continue this chapter in the next lecture. Allah Hafiz.